Imagine a living vertebrate whose heart stops entirely. For weeks, its lungs draw no breath, and its blood ceases to flow. Yet, it isn't dead. It survives by pumping its veins full of a natural, glucose-based antifreeze. This is the North American wood frog, known scientifically as Rana sylvatica. Found as far north as Alaska, this amphibian routinely endures temperatures plunging to -14 degrees Celsius. During these deep freezes, up to 70% of its total body water turns into solid ice. For a human, or almost any other vertebrate, a total halt of circulation and respiration, combined with frozen internal fluids, is the absolute clinical definition of death. The wood frog casually enters this state every winter and hops away in the spring. To understand how the frog pulls this off, we have to look at why freezing is lethal to living tissue. When water freezes, it expands and crystallizes. Inside a biological organism, these crystals act like microscopic daggers. As they grow, they puncture and shred delicate cell membranes from the inside out. Even if the sharp edges don't destroy the architecture of the cell, freezing causes a violent shift in osmotic pressure. As water between the cells freezes, it creates a concentration imbalance that aggressively sucks remaining liquid out of the surrounding cells, forcing them to collapse. To survive the winter, an organism has to engineer a way to simultaneously prevent the physical tearing of its tissue and the catastrophic collapse of its internal water balance. The wood frog's survival mechanism activates the moment frost touches its skin. This physical contact triggers an adrenaline-like nervous system response, kickstarting a massive breakdown of stored glycogen in the frotofree-gorged liver. This diagram illustrates the rapid glycogenolytic pathway inside liver cells. Enzymes like phosphorylase and phosphoglucomutase shear Sarge glycogen reserves down, yielding glucose 6-phosphate. Then, overexpressed bidirectional GLUT2 transporters act like open floodgates, pumping the newly synthesized glucose out of the liver and directly into the bloodstream. Within a few hours of ice forming on its skin, the glucose concentration in the frog's core organs increases by a factor of 100. A diabetic surge of this magnitude would easily kill a human. For the frog, this massive sugar spike acts as a colligative cryoprotectant. By densely packing its blood with dissolved glucose, it physically lowers the freezing point of its internal fluids. It is the exact same chemical principle as spreading salt on a snowy road. Lowering the freezing point buys time, but the frog doesn't actually avoid freezing, it manages it. Specialized ice-nucleating proteins deliberately trigger crystallization in the extracellular space, the area strictly outside the cells, at relatively high sub-zero temperatures. Once the freezing starts, a second set of molecules, called ice-binding proteins, attach directly to the growing edges of those extracellular ice crystals. By capping their expansion, these proteins limit the size of the crystals, ensuring they never grow large enough to shred the adjacent tissue. At the same time, water is deliberately drawn out of the cells by osmosis. The dense glucose and urea remain packed inside. The cell shrinks slightly, creating a safe physical buffer zone between its membrane and the sharp, controlled ice outside. By precisely controlling where ice forms, the frog's vital organs are entirely encased in solid ice without a single cell membrane rupturing. This document highlights the resulting physical state, a frozen wood frog buried in winter leaf litter. With up to 70% of its body fluid frozen solid, the animal enters a state of total ischemia. There is no breathing, no brain activity, and no heartbeat. Being deprived of oxygen this long usually leads to rapid tissue necrosis. The frog counters this with severe metabolic rate depression, practically halting its energy needs. Simultaneously, it maintains massive antioxidant defenses, using enzymes like superoxide dismutase and catalase to block oxidative damage while frozen. The ability to halt biological time and perfectly preserve living tissue without decay is exactly what modern medicine desperately needs. Today, medical researchers are actively studying the wood frog. By reverse engineering these specific glucose pathways and antifreeze proteins, they aim to solve one of the greatest challenges in healthcare: how to safely freeze, store, and transport fragile human organs for transplantation. When the environmental temperature finally rises in the spring, the physiological reboot sequence begins. The frog slowly thaws from the inside out. The massive concentration of glucose that kept the cells intact is rapidly cleared from the bloodstream. It is pumped back into the liver and reassembled into glycogen, stored and ready for the next winter. As the ice melts into liquid water, blood circulation resumes. Without any external electrical shock or stimulation, the dormant heart autonomously restarts its rhythm. Within 24 hours, the wood frog hops out of the leaf litter, entirely unharmed, carrying a living blueprint for the future of cryogenic medicine. Dive deeper into the hidden secrets that shape our reality. Click the subscribe button, activate the bell, and see you in the next scientific investigation, only on TEMBODocs.